Hey everybody, so I have the daily directions for April 28th as well as April 29th because I do have the STEM case that will probably take 2 days to get through, especially since this video is probably going to be a longer direction video as well as I'm going to do kind of the introduction guidance video on how to do the STEM case. After kind of going through some things and kind of think about it, I did decide to add an extra day onto the homeostasis lesson 7 to be able to kind of finish those Google Slides. I figured, about 15 minutes of the videos. So I do want to give you a little bit more time and some extra time to work on and finish that, or the Google Slides. So you can be able to get through that and basically take your time with it. So looking at is homeostasis lesson 8. So again, what we're going to be doing is we're going to be doing another STEM case through the Explorer Learning website. What we're going to be doing with this one is you're going to be basically acting as a veterinarian, right? And you're going to be helping a young calf named Clark who's having seizures. So what we need to try and do is we need to figure out, okay, why is this happening? So we're going to learn a little more about osmosis by flying into his brain and kind of seeing what is happening, and we're going to try and save Clark. As you can see down here is like I have the, watching this video to be able to make sure you understand. I'm going to walk through sections of it to kind of show what different parts mean. So all you need to do is click on the Gizmo case study for osmosis. It'll pop up. You can see here, right now I'm kind of listed as student X. So go to my classes, and then right here I have my third hour biology. And you can come down here to the osmosis high school STEM case. And so with this, and for me, I'm going to be clicking resume because I have started just a tiny bit, and it's actually the very beginning here because I didn't actually answer any of the questions. So here it starts out, says welcome to the osmosis STEM case. Click when ready. So again, we're going to be focused on, if you're ever unsure of what you need to do, look for anything that's kind of flashing blue. So you can see here around the click when ready, there's the kind of like blue flashing. That's kind of the indicator that that is the next part that you're going to want to go and you're going to be doing. So you're going to click here when you're ready. Here kind of here's Clark. He says you're a veterinarian. 5-day-old calf named Clark is brought to your clinic. So the first thing you do is review the symptoms. So here again, the client sent this cell phone video. You can see here where the video is flashing. It's a little bit hard to see. So sometimes we have to look a little bit for that. So we click on this, we can see now. So here's Clark, the calf, and what he's doing is he's having a seizure. Right? So then we kind of learn more about this. So you click next. So you see here symptoms. He's lying down. He's non-responsive. He's in a daze. He has diarrhea as well as he's seizuring. So again, just like the last STEM case for evolution, what you're going to do is you're going to click on the orange terms to get the glossary or kind of the definition. So you can see here diarrhea is a condition in which the organism's feces or its poop contains too much water. So what does it leads to a loss of water and salt from the body, and dehydration, low salting then can cause other issues. So I remember in class we were talking about this during our kind of the viruses bacteria unit. One thing as I mentioned is like diarrhea, one of the key things is maintaining like your water in your body. So drinking, basically any kind of thing that has high in water as well as high in kind of what we kind of consider our ions like salts and sugars. Another term you may want to look up is seizuring. So again, you want to look down and kind of see what a seizure actually is. Oh, not too far. So you can click on seizures, you can see a symptom where an animal shows abnormal electrical activity and the body normally shakes rapidly and uncontrollably. Okay. So those are kind of key symptoms. So you're just going to click next. So your goal is to find out what's causing seizures and pick the best treatment. So first we need to learn a little bit more about this. So it starts off with a handbook where it kind of goes through, and this is almost like the workbook part of this. So you use the arrows to navigate, and there's going to be different questions and parts that you have to do. And so some of this should be familiar. So we have been talking about like the solutes and the tendency last couple of days of the assignments we've done. So here again, seizures, if you need to, you can click on the orange link. So I'll bring up the glossary to learn a little bit more about what those terms are. All right, I would encourage you to make sure you read this because it'll make the answering the questions much easier as you go. So I'll click next. So first of all, learning like what diffusion is. So you can see, diffusion is moving particles from an area of high concentration to low concentration, basically until everything is equally spread out. So again, that's basic diffusion. Right, so now if we add in a cell membrane or a membrane of some type, right, with like here when we're looking at just oxygen, our oxygen particles are very, very small, right, and what they are is they can move across the membrane. That means they're permeable. Permeable means they can move through the membrane. So if we click play here, you can see the oxygen particles can make it through the membrane. So that means that it is permeable, right? And again, they're going to move until they're equal on both sides. Okay. And there, called the concentration gradient, it's going from high down to low. Okay. And here's, again, it's kind of tricky to see, but the flashing arrow. Okay. So here it says create a concentration gradient to make oxygen molecules move or diffuse across the membrane into the cell. So you need to figure out, you want to make it so you want to take oxygen particles from outside the cell into the cell. So you're going to be choosing 0 or 30 for both, one of them for outside the cell and one of them for inside the cell. Okay. And I'm going to pause the video here so that way I'll give you just give you the answer. Okay, so next part then is going to be looking at like ions. So one of the key things is, remember, the cell membrane is semi-permeable, so it's not going to let everything through. So like sodium ions, for example, get stuck and they actually can't travel through the cell membrane. So that means that the membrane is impermeable. Impermeable means it cannot diffuse across or through the membrane. Okay. So what about water? And water is going to be very similar. You can see here, water, as you click on it, water can't get through the membrane. So that means it's basically, again, it is impermeable. But one of those things is we know that water needs to get into the cells because our cells need water. So what we can do is we can add in pores or openings. So if we add pores into the membrane, and now that water can get through. But remember, the inside of the cell membrane doesn't like water, so it's not going to be able to be just pores that are going to be there. So we're going to have to add something in. And when, this is facilitated fusion. If it's facilitated, it means it's basically it's helping it happen. So what we add in are these molecules called aquaporins. So aqua just simply means water. Porins means pores. So basically they're openings that water molecules can move through. So click play, and this allows them to be able to make it through it. Okay. And again here, the ultimate goal of osmosis and diffusion and basically any type of this movement is equilibrium. Right? Again, equilibrium is basically when things are in balance or they're equally spread out. So that way they're both sides are equal to each other. Okay. So again, click down here. Again, this is one that's took me a minute. It's the first time I threw this, so I kind of look down here to see the arrow that's flashing. Okay. So again, it's talking about moving from high concentration to low concentrations. What happens with osmosis. Click next. So how do solutes affect this? So this is where we're going to be looking at what happens. So what water molecules do is, like, this is like a salt particle, so sodium chloride. So the water is going to actually like dissolve it or break it apart, and water is going to attach to those separate pieces. So what we have is now we have less what we call free water. Free water is water that is not attached to any other substance. Okay. Whereas the soluble bound water are things that are attached. So that's one of those key concepts here. Soluble bound water molecules cannot pass through the pores. Only free water molecules can go through aquaporins. So we're looking at those pores. Only the free water molecules that are not attached to anything can make it into the cell, where ones that are bound to either the chlorine or sodium get stuck on the outside or stuck on the inside, depending on where they are. Yeah. So again, here you look. So what do you predict will happen to the concentration of the free water molecules if you add salt? So if you add salt to the solution here, is the amount of free water molecules going to increase or will it decrease? And so I'll let you choose that. I'm going to kind of skip through this part to let you answer the question. Okay, so the next page then is going to make you kind of predict which direction is the water going to move. So this is similar to the hypertonic, hypotonic, isotonic slide assignment. So you're looking at the amount of water. So you can see there's 12 free water molecules on the outside, 32 free on the inside. So you're going to decide, okay, where does the water want to move? Does it want to go out of the cell? So is it going from inside out, or does it want to go into the cell? So based on the amount of free water, which direction do you think is it going to move? Okay, so the next part then relates directly to the assignment from Friday and Monday. So we're looking at the three types of what we call the tonicity, or they're hypertonic, hypotonic, or isotonic. So again, we're looking at kind of solutes. So remember, the solution is a hypertonic, right, when compared to the inside. So that means you can see it's hypertonic. Hyper simply means more. So a lot of times when people are hyper, they tend to have more energy. So you can see there's more of the bound water molecules on the outside than the inside. So the outside is hypertonic, the inside would be hypotonic. Right? So you can see here hypotonic, simply hypo means less. So there's less solutes or less water molecules bound. Right? If they're equal, or sorry, and this one shows the opposite. So if they show here we have, hypotonic is there's less outside, more inside. So the area where there's more, right, is hypo or hyper. As remember, hyper means more energy, or it's kind of easy way to think about it. If somebody's really hyper, they have more energy. So hyper simply means there's more molecules. Hypo means low or less, so there's less. All right. When they're equal, we call that isotonic. So iso basically means kind of equal. All right, so you kind of see how they are equal to each other here. They both have 5 bound water molecules on each side. Okay. So here you're going to make some predictions about what you think is going to happen. So if you drop red blood cells, if it's a hypertonic solution, so hypertonic means that there is more bound molecules. So you want to make a prediction of what you think is going to happen. All right, then there's going to be also one for isotonic and one for hypotonic. So I'll have you answer these and then be able to continue on. Okay, so next page again is continuing to look at the hypertonic, isotonic, and hypotonic. So what we're looking at here is like our blood. A lot of times we look at like the blood. So you can see here blood vessels. Right, that's what carries materials from one area to another. And so, and then we also have what we call the matrix. In this case, it's the brain matrix. But the matrix is normally kind of the area between cells or kind of the area surrounding. So we're kind of looking at the area that basically a lot of the materials is kind of floating around. So we're trying to compare here. So we're looking at this is basically, is there, in the image, is the blood hypertonic, isotonic, or hypotonic compared to the matrix of the brain? So again, you'll just simply choose one of these 3. Again, make your best guess, but what you think the answer is, and then you'll be able to submit it. Okay, so another key thing with this then is kind of like looking at pressure. So how is the pressure going to, or how is the movement of molecules going to affect the pressure? So the brain is inside the skull and can only basically swell a small amount. So if we have, what will happen to the pressure if water moves into it from the blood? So if we continue adding more and more basically water into our brain, what's going to happen to the kind of the pressure in there? So remember, the brain can only swell up so much because we have the rigid skull that's surrounding it. So is the pressure going to increase, decrease, or stay the same? So you can answer that, and then it'll kind of give you a definition, explanation for the answer. Okay, so the next part then says, what happens when pressure increases in the brain? So the brain contains billions of cells called neurons, which are basically just nerve cells, right? And they communicate with each other through electrical signals. So based off this, you're just going to take a guess here, and this one you probably won't know initially. All right, so if pressure increases, what's going to happen to the firing rate? So is it going to be something that's going to increase high? So with this, you can either grab and click on the slider or basically just click on the bar. So you're going to put what you think is going to happen to the firing rate or how often these cells are sending signals. All right, and then you simply will answer that, click next, and it'll be able to show you what happens. Okay, so that's kind of the walkthrough of the handbook or kind of the background or information on each of these. So looking at the osmosis, solutes, the different types of tonicity, and the pressure. So if you want to be able to, you can go through and review any of these that you would like to. Otherwise, you can go and do the actual case. So you click on the case right up here. All right, so what you're going to be doing now is we're actually going through and collecting data. All right, so I'm going to try and let you guys do this on your own. I know there's parts that can be confusing, but this video is starting to get a little bit longer than I had planned on. So if you have questions, please email me or let me know, and I will kind of help you as those questions pop up. All right, otherwise, good luck, and remember, you do have 2 days to do this, so don't feel like you're rushed to go through, and it will remember where you left off. Okay, so have a good day, and I'll see you guys again on, well, yeah, whatever.